Thank you, Lisette, for that wonderful presentation. As you said, we'll now be moving on to the Q&A portion of today's webinar. Many of you have already entered your questions, but if anyone else would like to ask Lisette a question, please enter it into the Q&A box now. Lisette, the first question is, how many times do you need to clean the probe in the acute preparation?
So you can clean the probes every day after the experiment, or you can clean them on different days. No, it doesn't matter. I mean, I recommend cleaning them frequently by submerging them in the enzymatic solution, because that will prolong the life of the probe.
Okay, thank you. Next question. How do you know the precise location of the probe in the brain?
Well, I mean, if you are working in the hippocampus, this is more or less direct, as I said, because we learn to infer the position from the precise position from the local field potentials and the information that we get from the recording. You can use a fluorescent dye to paint the back of the probe so that they leave a mark along the track. But you will never be able to identify precisely the tip of the probe, okay? So you will see where the shank was going through, following the track, but not so precisely. You need to learn. I recommend to learn to rely on different signals to try to localize yourself.
Okay. Next question. Are sights and LEDs functional all the time, or do they tend to fail over use?
Well, I mean, that may happen that, well, these probes are, the substrate is silicon, and therefore they are fragile, and you may touch one shank and break a shank. For instance, in our case, we have a couple of probes with only 3 shanks because we lose one of the shank, but you can still use it even if one shank is gone. Then they typically behave properly, but if you are not careful and you apply large currents, you can get trouble with the LED. In our experience, they work pretty well. We haven't seen any electrode or LED going away, except in one case, but that was something weird that happened with the passivation layer. So more or less they behave properly. Sometimes at the beginning you may have a failing electrode or a failing LED. That may happen, but basically when you buy the probe, they are tested for quality. So
okay. What is the maximum ripple frequency that is considered normal during a sharp wave?
That is a fantastic question. Ripple frequency is between 100 or 200 hertz, typically. But the more we play with the ripples, the more we learn that actually there is not a particular frequency. There is a range. And depending on how you identify ripples, ripples are typically detected by thresholding. So what you do is you estimate the amplitude in the 100 to 200 hertz, and then you look for the peaks. And that limits the number of events that you are recording. So the answer is that a normal ripple is between over 90 hertz to 200 hertz, but you can have slower and you may have some faster. And then pathological ripples tend to be faster, but not all the time. You also may have low frequency ripples. So you need to learn from a combination of features, whether it's normal or not.
Okay. How likely are you to disrupt/damage the surrounding cells when you dip the probes in the brain tissue?
Damage the surrounding. I mean, if you go carefully and you penetrate slowly, you don't damage that much. You see the cell. Typically, we recommend to penetrate slowly and to allow the tissue to stabilize. So never start recording immediately after penetration. Once you are in place and you are happy with location, you may allow some minutes, typically 15, 20 minutes, depending on how long you can wait in your preparation to stabilize. If you penetrate several times along the same place, then you damage. And you always leave a mark. It's not, I mean, if you are skilled, and Elena is very skilled in identifying the probe track, even if we don't use a dye. So that means that somehow you leave a mark, but the cells are healthy and you see appropriate firing rates in there.
Okay, thank you. Next question. Do you know how the impedances change after repeated acute use?
Well, impedance changes if you have the position of whatever biological material or whatever blood or whatever thing stick there. But as I said, if you clean the probe, it get back to normal and you see them working properly. So I recommend just cleaning them. You can also use a kind of agar phantom or agar block and insert the probe in the agar under the micro manipulator and visual control to remove all the dirty things. But as I said, if you take care of them properly, they work well. They work well.
Okay. When recording with micro LED probes, did you do any channel specific referencing?
Sorry, say it again.
Sure. When recording with micro LED probes, did you do any channel specific referencing?
No, we record against the ground, so we have the reference on the ground at the same connected. Yeah, we record against signals in the hippocampus are typically recorded against an occipital screw that we have implanted in the animal neck or over the occipital skull.
Okay. Is the plating procedure on the probe affecting in any way the LEDs?
The plating procedure, you mean gold plating? If it is gold plating, I mean, if that means, so we use the probe as we receive the probe from the production. So I don't know whether the question refers to the passivation layer or something. I don't know. Plating, we don't plate the electrodes after. So we use them as they are. They are very good signal to noise ratio. You don't really need to gold plate these electrodes. If I understood correctly the question.
Sure. To the attendee that asked that question, if you'd like to expand on that, please go ahead. Okay, next question is, say when you're recording neurons in the CA1 region, given that neurons are tightly packed with minimal empty space between two neurons, do these shanks rupture / penetrate any neurons?
If I say, if I saw what? I'm sorry, last part?
Sure. Do these shanks rupture / penetrate any neurons?
If
You
mean, I mean, no, I mean, you penetrate, maybe I don't understand the question, but no, you go blindly, you penetrate the region, you see them firing when you start reaching the stratus pyramidale, then they calm down, and then you collect the sharp wave, and then you know where you are, and there is not such a, during the acute recording, you don't see any particular change. There is some drift if the preparation is not stable enough, mechanically stable. That this may happen sometimes in the head fixed because you fix the skull, but the brain move within the skull, and then you may have. But I have to say that if you are carefully with the dura and you're careful enough with the fixation system, you don't have such a drift. And if you wait, in our hands, I mean, it's quite good. Over an hour, that's okay. More than an hour, you may have starting some drift, but KiloSort 2 can correct for that.
Okay. Next question is from Sam McKenzie. I have not seen such a strong cross-shank inhibition with a CAM-key promoter. How much of the inhibition do you think is due to direct stimulation of interneurons with your different opsin promoters?
Absolutely, yeah, exactly. This is, yeah, absolutely, Sam. I think that actually this is why we are still troubleshooting all these differences between the different mouse lines. And we think that on a specific opsin, there is some crosstalk in there that we are losing the specificity. Also, we think that somehow there is this inhibition because the microLED is, even if we go with a very low amplitude current, we saw huge responses. So I think that probably, yes, this is why we prefer to go with the cowbindin and the Taiwan to be more sparse in terms of the expression. You can also control expression by titering the titer of the diluting the titer of the adenovirus. But yes, I agree. I mean, there is some issues there that we are still troubleshooting and trying to understand. Yeah.
Next question. How big was your craniotomy and for how long was your recording stable?
So that's really depend on the experiment. I mean, you can be very, very tiny and open a very small craniotomy, which will be very, very good to facilitate recording stability, etc. But as you see, you have the shanks, the distance between the shank is 250 microns. That means that you need a craniotomy that accommodate the 4 shanks. If you lose a shank, because, for instance, in our case, we have a couple of probes in which we only have 3 shanks, and we like them, even with 3 shanks, because then we can do a smaller craniotomies. So you can adapt. This is why the head prep is good. Also, you can open several craniotomies. You can open one craniotomy for at the beginning of the week, and then on the middle of the week, you can move around. Yeah, you can control that.
Okay, next question is, which features do you use to differentiate physiological from pathological ripples if they are in the same frequency range?
Mm-hmm. Well, the first and most important is that whether the animal is epileptic or not. This is quite informative, but of course, this is a very stupid answer. So if you are blind to the animal, there are other features that allow you to know. For instance, in the epileptic animal, most of chart waves are very high amplitude. You have you have much more synchronization and synchronous firing. You have less specificity of firing during the ripples. You see more unit firing. If they are strongly pathological, you cannot sort properly, and this is a major problem because you have spike collisions, and the ripples are mini population spikes, and therefore you stop seeing the cell. And many people get confused and think that during these pathological events, neurons are silenced. What actually happens is that you cannot sort. So there are many features of the event that you can use. The amplitude of the sharp wave. We like to couple the recordings with linear probe recording to know also about the sharp wave at the stratus pyramidale, because this probe, they only target the stratus radiatum, because these probes are only targeting the stratus pyramidale. So a combination of features, amplitude, shapes, slopes, synchronization, etc.
Okay, Robert Aberkin asks, can you distinguish cowbindin positive pyramidal cell responses from cowbindin positive interneuron responses during optosimulation?
Yeah, great. Yes and no. Okay, I tell you. So you can use an adenovirus for the Cree with the double inverted oriented sequence with the CAMK2. But the problem in our hands is that this is expression is going to CA2. This is a nightmare. I mean, so we couldn't troubleshoot this way. So we are doing now is we are estimating how many cowbindin positive interneurons we have along the chance they are not quite populated. So we try to establish clear association between the interneurons in the neighborhood of the penetration to estimate whether we were targeting from them or not. But yes, this is another problem that we are trying to solve.
Okay, I think we have time for one last question. The attendee who asked about plating, I think resubmitted. Is there a need to plate the electrodes during different recordings and then they expand with gold or PDOT to reduce the impedance of the electrodes?
No, as I said, we don't have that need. As I said, if you take care of the probe, you clean them properly, and you use this enzymatic solution as I suggest, and you keep them cleaned, they are working as perfectly as the first day. The only reason why we changed the probe is because they broke. Not, to be honest. Actually, the head fixer prep is really, really good.
Okay, thank you. I think that's all our questions. Don't forget to, everyone, don't forget to register for next week's webinar in our series, Microcircuit Integration with Neuron Scale Optogenetics. You can find registration links at plexon.com and can contact Plexon at info@plexon.com if you have any questions about purchasing the NeuroLight optoelectrodes. Thank you to Lisette, and thank you to everyone who attended. We will be sending a follow-up email with the answers to the questions asked during the question and answer session sometime tomorrow, probably. So thank you for attending. That is the end of our webinar.
Thank you. Thank you.